Welcome to another lecture by MedicoMedics, Learning Made Easy. Cell Biology, Chapter 3, Cell Membranes. In this lecture, we will discuss the structure of the cell membrane, discuss transport mechanisms. We will discuss passive transport, diffusion, osmosis, and facilitated diffusion. We will compare it to active transport. We will discuss endocytosis and exocytosis, cell membranes, and signaling, and end with a summary. Now, what's the structure of the cell membrane? In the previous chapter, we mentioned that it has a phospholipid bilayer. Now, the phospholipid bilayer is composed of phospholipids with hydrophilic, meaning water-loving, heads, and hydrophobic, meaning water-fearing, tails. It is arranged in a bilayer to separate the internal and external environments. Furthermore, we have proteins. So integral proteins span the membrane, acting as channels or transporters. Furthermore, we have peripheral proteins attaching to the membrane's surface. These are aiding in the signaling or supporting. Another thing is cholesterol, which provides stability and flexibility to the membrane. We also have carbohydrates that are attached to proteins and lipids. These are involved in cell recognition and signaling. And here in this illustration, you can see these different parts. So we have the phospholipid bilayer. We have the head here and we have the tails here. And as you can see, tails from the membrane wall on the outside of the cell and the membrane wall on the inside of the cell are connecting. So this is why we call it a bilayer. And we learned here that these tails are hydrophobic, so they don't like water, while the heads on the outside and the heads on the inside are hydrophilic. Another thing we learned is that there are proteins creating channels for things entering from the extracellular environment and things from the intracellular environment to exit. Now, there are other structures like carbohydrates and lipids, etc., on these cell membranes, helping with recognizing things that are floating by, wanting to enter, for example. Now, transport mechanisms overview. When we talk about transport mechanisms, we have passive transport, where no energy is required, and we have active transport, where energy is required. Now, when we talk about passive transport, we talk about diffusion, osmosis, and facilitated diffusion. When we talk about active transport, we talk about protein pumps, or the process of endocytosis and exocytosis. So the main difference is that some substances will just sneak through rather easily through the processes of passive transport, and other substances need help or energy to pass the membrane. So let's dive deeper into passive transport first. So let's begin first with passive transport and the process of diffusion. Now, diffusion is the movement of molecules from an area of high concentration to low concentration. For example, the oxygen and carbon dioxide exchange in the lungs is an example of passive transport or diffusion. And again, no energy is required. So diffusion is like the scent of a perfume spreading across a room. It moves from where it's strongest to where it's weaker. Now let's look at a special kind of diffusion called osmosis. So another passive transport is osmosis. This is movement of water molecules through a semi-permeable membrane from low solute concentration to high solute concentration. This is very important in cells because it helps maintain fluid balance. Another passive transport is facilitated diffusion. where movement of molecules is down their concentration gradient with the help of a transport protein. So, for example, glucose transport into cells using carrier proteins. Again, no energy is required. So, facilitated diffusion is like using a key card to enter a secure building. You still follow the rules, but you need special access. Now, what's important here is that we said the movement of molecules down their concentration gradient, right? Now, it is facilitated with the help of a transport protein. But what if we come across when the cell needs to move substances against the gradient? So when there is opposition and it's not enough to diffuse through the membrane, even with the help of transport proteins or by osmosis, for example. This is where we need to talk about active transport. So in active transport, we need energy to facilitate movement. And it moves substances against their concentration gradient. So for example, the sodium-potassium pump functions in this way. Another one is calcium pumps in muscle cells, for example. So active transport is like, imagine you're pushing a ball uphill, right? It takes extra energy to move things when gravity is pulling it back down. So this is where the role of ATP, and if you remember previous lectures, the main organelle involved in the production of ATP is occurring in the mitochondria of the cell. Two other important concepts are endocytosis and exocytosis. So endo means essentially in, while exo means out or exit. So endocytosis is the process by which cells engulf substances into vesicles. An example of this is phagocytosis, when white blood cells are eating bacteria. So you can see here in this illustration, endocytosis, right? Here is the bacteria, and it's being engulfed. It's entering into the macrophage, or the neutrophil in this case. And following along, it then degrades, or the bacteria is broken down. And when that is finished, it then undergoes exocytosis. So exocytosis then is the process by which cells expel substances in vesicles. Example is the release of neurotransmitters in nerve cells. So just remember, endo, into, exo, or exit is out from. Now let's discuss cell membranes and signaling. So we mentioned that there are on the cell membrane proteins, for example, that are receptors, and they are on the membrane detecting chemical signals. So for example, hormones or neurotransmitters. What then occurs is signal transduction. So signal binding triggers a response inside the cell. So here is an illustration whereby a ligand, which can be any molecule, here in blue, is binding to a receptor on the cell membrane. When it's binded, there is a relay, right? Signal transduction is happening, arriving into the cell, causing or creating a response. Examples of this in medicine could be insulin receptors for glucose uptake. Targeted drugs that block specific receptors in cancer therapy is another example in medicine where this is actually quite crucial. So what we do then is we take some drug to block this receptor so it doesn't bind here. Because let's say we find out that if this thing floating around binds to a receptor on our cell membrane, it will relay a signal leading to, I don't know, the production of something else, right? And we don't want this something else to happen. And since we probably can't get inside to kill these off, what we do is we say, okay, we know that this receptor is binding these. We don't want that to happen. So we will give a drug that will go and block this receptor. And then we will not have this response happening. So that's an example of targeted drugs that block specific receptors. And so the membranes of our cells act as well as antennas. They pick up signals and then pass them inside the cell to trigger actions. In summary then, the cell membrane is a dynamic structure vital for protection, transport, and communication. Transport mechanisms, passive and active, ensure the cell maintains balance. And if you remember, we had passive transport like osmosis, facilitated diffusion, and diffusion. When we talk about active transport, what is absolutely crucial is to remember the use and need of ATP to make it happen. And active kind of implies that activity is involved, right? And you can remember the mountain metaphor, whereby you're trying to push something uphill. You will have resistance, right? So you need to create and use energy to be able to push against this gradient that is pushing down. This is not happening when we talk about passive transport. And finally then, membranes enable cells to respond to signals and interact with their environment. Now with this foundation, we're ready to explore how cells communicate with each other in cell signaling next. So we have reached the end of chapter 3. Continue now to chapter 4.